Good morning children, welcome to NEET AIPMT Foundation Biology session. You are watching plant breeding and crop improvement part 3. Children, we have been discussing from the past 2 sessions about the plant breeding. What is the necessity of this plant breeding? What is plant breeding, children? Plant breeding is the genetic improvement of the crops. For what purpose? In order to create desired varieties of plant types and that of the better suited for cultivation, giving better yields and resistant to diseases. Okay. So, in the today's session we are going to discuss about the hybridization. Okay. So, before going to that, let us have some discuss some few facts. The development of several high yielding varieties of wheat and rice in the mid 1960s as a result of various plant breeding techniques led to dramatic increase in food production in our country. So, that phase is often referred to as green revolution. Okay. So, who is father of green revolution? M.S. Swaminathan. Okay. In this green revolution, some Indian hybrid crops such as wheat, rice, sugarcane and millets, the food crops were developed during that period 1960s. Okay. In 1963, several wheat varieties such as Sonalika and Kalyan Sona, which were high yielding and disease resistant, were introduced all over the wheat growing belt of India. The semi dwarf varieties derived from IR8 and Taichung native 1 were introduced in 1966. Later, the better yielding semi dwarf varieties of Jaya and Ratna were developed in India. Okay. So, all these are possible, by using this the science and technology, the knowledge in this crop improvement, plant breeding is useful. Okay. High yielding and disease resistant varieties, okay, in wheat as well as sugar cane. Okay. So, if you take South India, Saccharum officinarum grown in the South India having thicker stems and higher sugar content. To obtain the varieties having high yield, thick stems with high sugar content and ability to grow in sugarcane areas of North India. Okay. So, how it is possible? Due to this hybridization technique. Okay, let us go for hybridization technique today. Are you ready, children? What is hybridization? Hybridization is the mixing up of characters of two parents in the new offsprings. Okay, previously in the previous session we have studied about the selection. Okay, compared to selection, this hybridization has got a greater advantage. Okay. So, it is done, hybridization is done between the two varieties of same species, that may be intraspecific within the same species or between the two different species, that is interspecific. This is, please make a correction here, interspecific, ER. Okay. Intraspecific and interspecific techniques, hybridizations. The first generation progeny of hybrid are called as segregates. The types of hybridization: on the basis of the nature of crop plants, crossed hybridization can be of the following types: a cross between two plants belonging to the same species, it is called as intravarietal hybridization or intraspecific hybridization. Intra means within. The second one is the cross between two plants belonging to different varieties. Okay. So, in between, within. Okay, in between means different varieties. It is called intervarietal hybridization. Okay. So, when a hybrid is produced by fusion of somatic cells of two varieties of species, it is known as somatic hybridization or somatic hybrid. Okay. The process of producing somatic hybrids is called as somatic hybridization. Okay, children. Now, the third one, that is a cross between the two plants belonging to the same species, called intraspecific. Okay. Intravarietal hybridization, intervarietal hybridization and intraspecific hybridization. Okay. Number 4, a cross between the two plants belonging to two different species, that is interspecific hybridization, and a cross between the two plants belonging to two different genera. There we come across species. Now, two different genera, it is called as intergeneric hybridization. Intergeneric hybridization. So, what are the aims of hybridization? For the plant breeding, different aims of hybridization are as follows. The first one, to produce new varieties of crop plants. Number 2, to bring about the variations in the particular group of plants. Number 3, to combine the good characters of two different plants in a single plant. And number 4, to exploit the principle of hybrid vigor to produce a plant variety which is superior to both the parents. What is hybrid vigor means? The superiority of the offsprings over their parents is called as a hybrid vigor. Okay. So, the plant breeding, especially the hybridization, has several advantages. The first one, to produce new varieties of crop plants. Okay. The second one, to bring about variations. Variations means the changes, differences, more good qualities, desired qualities. And the third one, to combine good characters of different plants in a single parent. Okay. For example, some desired characters like dwarf plant, high yielding plant, okay, and less crop duration, these three are there. These three qualities you can produce in a single plant by this hybridization technique. Next, to exploit principle of hybrid vigor, okay, the superiority of the offsprings over its parents, to produce a plant variety with superior, when both the parents are there, one parent is having some qualities, another parent is having some qualities, but the offspring which is produced by crossing those two parents may show hybrid vigor. Okay. So, these are the advantages, and more advantages are there with this hybridization. What is the process of hybridization? The actual process of hybridization or plant breeding, hybrid plant breeding, includes some phases. The first one, selection of parents, selection of parent plants. The second one is selfing of the parents, okay, selfing of parent plants. Next, method of hybridization. The next one is harvesting of hybrid seeds. Okay. So, these are the 4 phases involved in any hybridization process. Selection of parent plants, the plants with good desired qualities we have to select. Then selfing of the plants means self pollination. The next, method of hybridization, we have to select the method. It may be intravarietal or intervarietal or interspecific or intergeneric, whatever the method of hybridization which is suitable, which is advantageous for that particular species, we have to select. Then after hybridization, After the production, harvesting of the hybrid seeds is most important. Okay. So, first phase, selection of parent plants. The parent crop plants are selected in such a way that it is ensured that they are healthy, they should be pure, that is they are not contaminated with the foreign pollen grains. Okay. Such a type of plants, it is the first step and most important step. The result of the hybridization depends upon this step only, the selection of the parent plants. Okay. Pure breed plants we have to select. Then the desired characters must be present in those plants. Desired characters means the characters like okay, high yielding, disease resistant, less crop duration, these are all the desired characters. Okay. The parent plant should possess these qualities. Okay. Next, in that selection of the parents, the process forceps method. Okay, this forceps method involves the process, the removal of stamens from an intact flower with the help of a pair of forceps. You know, forceps means like a scissor only. Okay. So, with the help of that forceps, we have to remove the stamens from an intact flower. So, this is carried out in large flowers like paddy, pea. Why to remove these stamens means? Because in order to get the result, we need to allow the parents to undergo cross pollination. Okay. To avoid that self pollination, the stamens must be removed in one of the parent. Then how? Next one is hot water treatment. So, in case of small flowers, for example, in large flowers this forceps method is useful, but in case of the small flowers like millets, sorghum, okay, the stamens are too small to be removed with the help of forceps. So, hence the panicle is dipped in warm water. The panicle means, that is inflorescence, the structure with the flowers is dipped in warm water at 45 degrees centigrade for how many minutes? For 10 minutes. So, which inactivates the anthers, Next, third step, male sterility method. Another method, that is male sterility method. In plants like barley, onion, the male sterile lines are used. Okay. Those sterile in nature and cannot produce the functional pollen grains. So, by using this male sterile method, sterile lines, so the flowers become sterile and they will not produce the pollen grains, male gametes. Okay. The male sterile lines are obtained by the spraying of some chemicals, that is called artificial phytohormones. Okay. Some hormones can be sprayed so that the male flowers become sterile, means the male parts of the flower like stamens become the sterile and they will not produce the functional pollen grains. Okay. This is the another method, male sterility method. And next, to avoid this self pollination, the bagging can be done. Okay. The male or female plants are covered with cellophane or polythene or paper bags to prevent the contamination by foreign pollen grains. Okay. The foreign pollen grains means other plants may be there which are having the non desired characters, poor qualities like tall plant, less yield. Okay. The pollen grains belongs to same plant, same species, but showing different desirable characters. So, to avoid the contamination with the foreign pollen grains in the male plant, the cross pollination in the female plants, this bagging can be done. Okay. So, these bags are made porous. Why? For the purpose of aeration. The bags in case of male plants are removed after the pollen grains are shedded off and used for artificial pollination. Okay, we have to use those pollen grains of the desired plant only with the desired qualities to pollinate. Okay. So, that is called artificial pollination, but in case of female plants, the bags are kept for a long period because to avoid the contamination. Next, crossing. The third phase is crossing. So, after the anthers get matured, they shed off the pollen grains in the male plant. So, these are collected in cellophane bags or in petri plates and are rubbed on the stigma of a emasculated or bagged female plant by removing the bag. Okay, that is called emasculation method. Okay. We have to collect the good pollen after the maturation of anther of the male plant. Then these are collected in the cellophane bags. Cellophane is the material. Okay, the bags made with that material. We have to collect the pollen grain, or we can also collect the pollen grains in the petri plates, and those pollens should be, okay, the pollen, the powdery like male gametes it is. It should be rubbed on the stigma of emasculated female plant. Okay. Before rubbing, we have to remove the bag. So, this is called artificial cross pollination. After crossing, the bag is again tied and maintained till the seed setting is completed. Okay. So, then the pollination, fertilization, and it will continue the female flowers. Okay. We have to cover the bag until the seed formation, seed setting is completed. The process of crossing is mostly carried out in the morning hours, when the stigma is receptive towards pollen grains. Okay. In the morning hours, the stigma is sticky and it is ready for receiving the pollen grains. That is why the artificial cross pollination can be done during the morning hours only. Okay. The next fourth step is labeling. The labeling is done for the plant breeding with a tag attached to the crossed plant. Okay. Experimental plant, the plant which is the hybridization process is done, for that plant we have to tag. It should contain the relevant information like details of the parent plants, date of emasculation, date of bagging and crossing, okay, field reference number, etc. All these things should be labeled. In that label we have to mention. Harvesting of hybrid seeds. Then after the seed setting, the hybrid seeds are formed. The last phase is harvesting of hybrid seeds. So, after the seeds are matured, the bags are removed, the seeds are harvested and collected in a labeled envelope. So, they are preserved carefully and used in the next growing season for hybrid plant breeding. Okay, this is the method of production of hybrid seeds. Okay. Timely harvesting is important, preserving is important. Okay. So, this is the technique of hybridization. Okay. Hope you followed, children. Read the book. Thank you for watching our video. Please subscribe our channel and press the bell icon for receiving latest updates.